Yeah, so I'm Darcy Peterka. I'm a senior scientist here at the Zuckerman Institute at Columbia University. It's kind of what we say is a multidisciplinary institute aimed at unraveling some of the mysteries of the mind and brain. It's mainly neuroscientists, but there's a handful of engineers, people in statistics, electrical engineering, and so on. And we all kind of work together to try and solve some of these problems. My kind of, let's say, forte is developing optical tools for reading and writing activity in the brain and trying to apply these for understanding particular computational problems. Most of what I'll talk about, I'll kind of get it out early that, let's see if this flips over. I should say that, many of the things I'm talking about here a lot of the work I'd say a contributor on, but a lot of this stuff is also work that's primarily done by others that I have the privilege of collaborating with. So I want to just highlight them early. And I've had these long-standing collaborations with Liam Paninsky, who develops algorithms for extracting activity, and some of his team, Ian Kinsella, Amal Harsakar, Melissa Wu, and Marcus Triplett, working on kind of different aspects of signal extraction and optimization problems for kind of online experiments. John Cunningham's also in statistics, and with Taiga, he's been working on these AWS, essentially instantiations, so their code can kind of be open to the world and runnable by anybody. I have long collaborated with Rui Costa, who is here now, but will be joining Jerome and Caspar at the Allen Institute starting this summer as president of the Allen. He'll maintain a lab here, so we still will have the ongoing collaborations. And Alice, Vivek, and Inej have been instrumental in some of the work that I'll show. And then there's me, as I'm kind of hanging out on the side, just like I usually am. And then there's Luke Hammond, who's a colleague of mine, kind of within the cellular imaging infrastructure here, who develops a lot of tools for kind of pipelines for analyzing brain and neural circuits using open source tools and kind of using AI segmentation. So, when I show the slides, almost all the slides I'll show will have contributions from these members, and I'll highlight them when I can and when I remember. So I wanted to get it out of the way first. What we're all really trying to do is link activity to behavior. So many of us are familiar with things like this. This is a mouse that it's trained to lick when it sees vertical bars. You see it's patient. It doesn't do anything. It's got a vertical bar, and now you'll see its tongue move like crazy. It gets a lick. It's pretty well trained at this point. It knows to lick. It withholds on other tasks. And this is kind of cool. This shows that we can get mice to learn tasks pretty well. And the thing that's kind of nice about it now is, beyond just learning the tasks now we can image the activity in their brain. So this is looking at GCamp in mouse V1 while the mouse is doing one of these tasks, watching a movie. I don't show it linked because it doesn't really matter what it's doing, but we get something like this. We get a lot of cool activity, and it's really awesome to see. You really get the feeling that you're kind of watching the mouse thinking or doing some kind of computation. But the real challenge is this is kind of great and all, but what does it mean? So we see this activity, and now we have to somehow find ways to image enough, regress it against the behavior that we're monitoring, and try and take into account all the kind of states that we're not monitoring, and then figure out which neurons are important. And many times you can find neurons that have correlations with your task, but you're always questioning, are they causal? Are they necessary for the task? Are they sufficient? And how they're governed. And this has been very hard, and one reason it's been very hard is because in most cases, we're looking at only a very small patch of neurons, a small amount of activity over a limited time window. So there's a lot of things that we just don't see. So what's one of the constraints is our essentially inability to record from enough neurons pretty quickly. So there's been a lot of work in the last year by Spencer has been one. Jerome when he was with Mark, and now at the Allen has made some big instruments, but many others now have these larger field of view microscopes that can image kind of fairly large areas with high SNR at moderate speeds, I would say, not yet high speeds. So this has been easy. I mean, this is kind of sampling of the field of view sizes respectively embedded in these systems. And as the field of view goes up and we still want high resolution, the frame rates goes down. And it's still really hard to image large areas with high speed and high resolution. It's a real challenge. So, why inherently are these things difficult with most of our kinds of microscopy? And we've heard a lot about it. I'm also privileged to go last year because there's been so many great talks that have been bringing up all of these points. So, I think by now you know this, but multi-photon images are made out of pixels and voxels. If you want a certain number of pixels at a certain frame rate, you get a pixel rate. And if those pixels are kind of you want a lot of them, the time per pixel, average time you can spend, is low. Unfortunately, the signal is proportional to the dwell time. So the signal is proportional to essentially the fluorescence the instantaneous fluorescence and essentially the amount of time you spend on it. And what you see here is a familiar 1-photon versus 2-photon image. And you see then the 2-photon image on the little dot kind of in the center. It generates a point. It's essentially a point sampler. So one of the questions is, first, do we need all of these pixels? And do all the pixels really need to look like this? So when I say where the pixel is, right, this is the point spread function of the microscope I'm talking about under 2-photon excitation. So this is a volume of tissue or fluorophores or whatever that's excited by the 2-photon microscope, and it generates a fluorescent signal that you can then record on your photomultiplier tubes and capture images. And in most cases, we like having this small point spread function because it gives us very high resolution. If you think of your kind of a statistical distribution and how would you sample that, this is kind of like a delta function sampler. Wherever I put this PSF, this delta function, I get a sample of what's under the spot and nowhere else. This can be great, but and this is great if you really need to generate a very high resolution spatial image. But sometimes we have to think about more broadly and say, well, is our purpose for doing this imaging generating a high resolution image? What is our purpose? What's our goal of these experiments? And sometimes we have mixed goals, but sometimes these goals can be split into multiple tasks. So you could get a high resolution image one time, and then you could get a fast functional image at the other time. And what I think many of the people have described it talking before me is you can really, you need to think about the task, and you can kind of change this point spread function maybe, or change your microscope in your system to get more information. So what do we do to kind of increase this information? Well, one, if we want to reduce the number of pixels but still get the information, we want to increase the nominal information per pixel. And the way that we usually do these things is with projections and with priors. So we design our systems, and Nico described this very well. We kind of jointly design our strategies based on the task. And these can be very complex kind of computational strategies, like in lensless imaging is an example where the patterns are so far from human interpretable that the raw image is literally meaningless without having the strong priors or some way to extract it, to something that looks maybe a little bit different, like what I'll show later, maybe a Bessel beam image, where we project along an axial line and you get an image that looks kind of like the Max Z projection. We took this approach and we said, well, why can we even do this kind of stuff? And I think we talked about it in these previous talks. Well, neural activity is generally spatiotemporally sparse. That doesn't mean it's not complex, but it's sparse. So what we're looking at here is just, this was an ETL stepped image, looking at a few different planes in visual cortex, while mouse was basically just like watching a random movie. And these are just taken to show, and it's sped up a little bit to show the repertoire of activity. And in each of the frames, we see, the neurons, when they go on, they're bright and there's a bit of a background. Well, I can just do literally raw arithmetic. So this isn't simultaneously captured. I'm literally in the bottom panel. I'm taking the frames for each of the top panels, and I'm just going in image J, and I'm adding these movies together. I'm doing a linear summation, just pure arithmetic. And what you see with one plane, I could add a second plane, third plane, and even the fourth plane, the one that would be on the, let's say, this bottom right here, even though the background is higher, you generally see individual neurons pretty well. And with that basically means that like I might have some hope of extracting, if I can take advantage of these characteristics, some way of both imaging these things simultaneously and extracting out the signals in a faithful way. So the way that we do it, you've heard a lot about it already here. We use these Spatialite Modulators. These are, in this case, we use those liquid crystal devices that generate an arbitrary phase pattern that then can direct a beam. So if the kind of phase pattern is flat, we just got to point on the objective. If we put like a prism ramp, this would be a phase wrapped version, the mod 2 pi. So instead of a ramp, it looks like these steps. We can shift the beam laterally, and the others did great jobs of explaining the kind of the Fourier domain calculus that can go into doing these displacements. Well, you can do multiple spots. You can put lens functions on. You can make 2 spots, 3 spots, everything in kind of 3D. And this works pretty well, especially if you use some of the modern tools for generating algorithms that Nico just described. So the other thing that you can do is these things are software, so you can really actually switch them really quickly. So this is an 8th speed modulation on this thing. It's normally changed every 3 milliseconds. This is 64 speed. So this is looking at a uniform fluorescent slab, like a piece of fluorescent plastic, where I'm using an SLM to multiplex a beam and shine it into multiple spots at once. So not only can we kind of direct a beam anywhere we want flexibly without moving anything physical except for the liquid crystals in this device, we can change it dynamically. The thing that you should note that this image and these tests were taken with kind of uncompensated holograms, and Kaspar talked about this, and Nico as well. These holograms SLMs are diffractive devices, so you have to use proper considerations like do power compensation to get the things to be homogeneous and so on. So don't take the random kind of power levels on the left side of the different spots as anything other than a complication that needs thought and correction to do it. So and then that little speckle pattern that you see illustrated right up here, this is just kind of the diffraction efficiency of our given SLM as we map it out in real life. And that's because we can model this theoretically, but of course, reality doesn't always match our theoretical dreams. So we actually measure this for each SLM we have, and we can characterize it and then correct for many of these things going on. So what we did with these things with work that I did when I was with Rafa Yusta as a kind of senior or kind of associate in his lab, and with a postdoc Wei Zhen that I helped mentor. He's now a professor at UC Davis. We made this as multi-beamlet configuration using SLM. So now you've seen many instantiations of these kind of microscopes in these drawings. You have a laser, something for modulating the power, and then basically an SLM, some beam shaping, an SLM, and then a telescope to relay it to the right place. So you have this, and then the rest is a microscope. This would be super easy for Bruno to put together, super easy for any of you to put it together, really. It's really the cost of the items that's the hard part. And what this can do is now he can use a Spatialite modulator to project multiple spots in space simultaneously, in this case axially, and these get scanned both simultaneously. So now you're taking an image where you're scanning multiple spots simultaneously. And if you look at the little figure, you can kind of imagine what goes on. If you have a sample that has 2 different structures at 2 different planes, this structure A and the structure B, because we're not temporally multiplexing the beam, we're only spatially modulating the beam, and the speed of light is essentially infinite relative to our Galvo and our system. What we have is we see essentially fluorescent signals from both of these components that superimpose on the collected image. So we get an image that looks a little bit worse. And maybe you could read A and B together, maybe you can't, but it's easy if you have the prior. If you know what A is and you know what B is, it's easy. If you don't, it might be more work. So what we showed in this paper, this is from a while ago now, is we could basically take multiple planes, we could make multiple spots and separate them in space and scan them together, and then get up essentially images that were the combination of the signals from both these planes. What you see here is just a max kind of a standard deviation projection of a time series of 2 different, the activities of neurons over 2 different planes taken sequentially. So this is again like that example in the first where it's just, these are just completely separate images. I took the 170 micron plane, the layer, basically layer 2, 3, and the 500 micron plane at separate times. If I sum them together, I just get something that looks a little bit messy. But now, instead of summing them together, I just basically use my Spatialite modulator and instead shine light onto both spots simultaneously, scan them together. I can kind of directly get this image directly, right? And you can see it's not super complicated, but there are definitely some overlapping signals, especially if we look at some areas here. It's hard for me to see everything on my little screen, but in many places, there's neurons that are on top of one another and overlapping. So this would be hard if you just had to circle ROIs and extract the traces. It's almost impossible, right? Like it would be very hard to get a faithful ROI. But we have these methods that we can essentially extract out these planes automatically, and Andrea talked about a lot of them using non-negative matrix factorization. So that's kind of what we did in this paper, and what we showed is, in this case, you can actually extract out signals. The left side is sequential imaging, the right side is simultaneous imaging. That the signals look great, there don't seem to be contaminated on all this other stuff. So it works really well. So this tells us that we can start putting essentially more information into each pixel that you record on the camera or the PMT system, and we can use some computational methods to extract it out. Because I think the NMF problem is really important, I'm going to go through this. I'm going to go through it probably quickly because Andrea had, but I think it's worth remembering for a lot of these things. So if we have two of these sources, sorry, I guess my slide format is messed up, but what we do is we end up unrolling these slides. This is a kind of pixel and timestamp. This is just an unrolled image, meaning instead of showing it as an XY format, we just take the first row and add the second row directly, to basically linearize it and make it just a long column. So what the column is all the pixels in the image, and what the columns are is a timestamp. And like Andrea showed, we can vectorize this image, and we can actually just factorize this as a spatial matrix and a temporal matrix. And all of the properties that Andrea showed are it's the same. I'm just going through this for, I think it's a very important concept, so it's good to know that this temporal matrix, a row of it is basically the independent activity of that source, which could be, let's say, a cell. The column of the spatial matrix A, it's a spatial layout of each source. So that's basically saying a source isn't everywhere in space. This is so, right? Neurons don't cover the whole image usually, they cover only a limited part. So this this column has mainly zeros in it and then some non-zero numbers that reflect the kind of the position of this layout. But if we look across the spatial matrix of A, if we look at the rows now, what that says is for a given pixel, how many different sources contribute to that input. So that's essentially saying I might have a pixel and two neurons might overlap because now I have this projector where I'm projecting from two planes together and they could just happen to overlap. So, you can model this out. You have this problem, F equals AC, you add some noise and you can do these observations. So you do this work and you can separate out these independent sources very, very well. So this was kind of one of the early instantiations of it. And now there's many, many variants. The one that's I think both most popular and featured would be like Cayman and now Viola from Andrea's lab, but there's also Sweet 2P, all these other things. So the important thing on these is I'll talk a little bit more about it, is that these methods now allow us to essentially jam more information in the pixel with very little actually prior knowledge. We're not putting big models on. All we're just saying is that we think at some point neurons are somewhat compact in space, and we think there's probably at least one time during our recording that they're not all simultaneously active. So those are the kind of constraints that lead to some of these good things. So this allows us to do a lot of cool stuff as optical methods developers, and you've heard a lot about this kind of thing, which is if we look at basically what gives rise to the fluorescence in a given pixel, the total signal, it has a bunch of microscope dependent components like the collection efficiency and so on. And then it has some things that nature sets, the fluorescence quantum yield, the two-photon cross-section. But this term that's basically in the integral here, this is whatever the concentration of what you're probing is times the 3D distribution of light integrated on the spatial temporal volume. So nature controls kind of this. We control this as microscopists. We can control the spatiotemporal distributions of light, right? Nico just gave beautiful examples of this. Casper gave examples. Everybody gave examples of how we can control the distribution of light in ways that aren't just this simple kind of little point, right? And there's been a lot of work in these areas now. And, this is an older slide, so I'll just highlight some, there was using Bessel beam work that, that was, around for a long time, but Na really made some useful instantiation for neuroscience and kind of made them very popular again, the way you extend the range in a long direction. So compared to normal beam, you do a projection along a kind of a long axis. There's our paper where we did spots, multiple spots, and you can do a lot of spots. It's not just 2 or 3. David Tank had this V-twins microscopy, and there's many, many other instantiations, each with a kind of a different advantage in a different instance. But the key is all of these and sample these projections. They have a different sampler of the tissue in ways that lump signal together in different ways that can be paired with an algorithm to extract out the data. And the way I like to think about it and the way I like to think about SLMs is like, I like to think about what almost all we do in imagers is we control the sparsity. We know if all the signals are very, very mixed, it's very, very difficult to extract things out. Essentially, we need to make things less complicated. So in two-photon microscopy, we do that, right? We have a little point normally. We try and make that PSF as good as possible. It samples something small, and it moves around kind of relatively slowly. So that's great. But most of the times we know our neural signals don't require that level of sparsity. We can have some intermediate levels. So now what we do is we change a point spread function in ways where we can control this, and I'll put more information back into our pixels, and then we can use smart algorithms to extract it. So I think the best choice of one of these things depends really on the nature of what you hope to collect, what your task is, and the nature of the tissue where you're trying to do your collection. And I should say that this is using just spatial multiplexing, and you can of course add these with temporal multiplexing, and many people have done this where you then split the beam temporally as well, and you can use temporal decoding strategies to get it out. And the most powerful systems, I think, use both, right? They do these spatial beam shaping and temporal beam shaping, and that's probably, in terms of all the microscopes that you've seen, I think kind of the SLAP 2, this microscope is probably I think like one of these kind of pinnacle designs. They had a very specific task that they wanted to solve. They thought about all the strategies of how to make it work, and they built a system that is very, very good at the problem they want to solve. So it's kind of focused engineering, smart, clever optical design, and a combination of the algorithms and understanding kind of the landscape of what they want to measure that can kind of produce these kind of instruments. So I want you all to think, as part of the workshop, is to think about when people really see these microscopes and, oh, this is the best thing since sliced bread. This does 7 trillion neurons. This does 1 neuron, but at a gigahertz, or this, whatever. You kind of can evaluate all of these claims based on what it is that you want to solve, the problem that you want to solve, and then start exploring these. And, I think all of us here at the workshop, we're happy to talk about it if people ping us with questions like, hey, what do you think would work, right? As developers, we really want people to use the best tools and the appropriate tools. And if that tool isn't mine, I'm not going to point you to my tool, right? Because that would be dumb. It would be dumb for me and it would be dumb for you. We'll point you to our other people's tools. So like we all are aware of what's going on in the landscape, and you've probably heard all of us talk about how happy we are as speakers to see each other's work, because that's how we learn what the best tools are, so we can point people to the real thing. So that's I think a important thing to know. So, fundamentally, how much control do we have over these distributions? Nico highlighted some of it, Casper as well, right? You have an energy, you have a rep rate of a laser, you have some max power that you put in. As the rep rate goes up, the peak power, the peak energy per pulse goes down, but before you can even hit your power level at low rep rate, you blow up your cells, you ablate them, it ionizes your cell, turns into a plasma, and it kills it. Which is great if you want to kill a specific cell, but not so great if you want to measure its activity. But long before that, usually you get saturation. And that means the light intensity is so strong that every fluorophore that's in that point spread function volume that could be excited gets excited. There's no point in increasing the power. But the way you can get around this then is if you can change the area of the beam, the spatial footprint, again, this kind of this integral I was talking about, now you can distribute the light over more places and the saturation limits go down this area. So you can now start getting more and more kind of useful signal before you hit the ablation limit. And there's things about lifetime, and I have a fancy plot, but I still like this handwritten plot because it just illustrates, like, we don't know what these values are in a lot of cases. So we need to get good data to constrain these kind of things, to figure it out. And I say good data and even bad data help define these constraints. So like, if you think you hit a limit, it's kind of good to share that, because then we as a field can figure out, like, is it a real limit or is it a limit in this instantiation? The other thing I wanted to say about this, and this was, this is a favorite book of mine as a book plug. It's Building Electro-Optical Systems, Making It All Work. So this guy is really great. I think it has a lot of really useful information about even alignment, estimating photon budgets, design constraints. It's kind of more set up for industry than academic research, but it's a fantastic book. And then, what I want to say is about this kind of thing is, when we build our microscopes, this is a quote, I put a little emphasis in there, but photons are like money. A certain number are needed for the job at hand, and they're easier to lose than to gain back. The idea of a budget applies to photons as to finances, but it is more complicated that not all photons are useful, as though we had to budget a mixture of green and purple dollars. Also, like the other kind, people sometimes don't even expect to be on budget. They settle for it. This was the best we could do, but I'm not sure what the problem was, right? And I think this is a great quote. And, I was very pleased in Kaspar's presentation, right? He showed apparent bleaching of the new glue sniffer variant, but then he showed that the SNR doesn't change at all, right? So this is a perfect example of these green and purple dollars, or green and purple photons, right? Some of the photons that came, they came to a system, you collected them, but they weren't useful photons. In some sense, the faster they bleach almost, if that bleaches faster than the glue sniffer, the better it is, because these are just like extra photons that you can't budget for in kind of good ways. So this really talks about like how to do it. The green photons are maybe the ones you want to collect, and the purple ones are the ones you want to avoid. So what you want to do is you want to collect as many of the good photons as you can while excluding all the non-modulated signals, like tissue fluorescence or autofocus fluorescence and so on like that. And that's why we still like 2p over 1p in many cases, is because we don't want those lots of background photons from 1p. They're a little bit harder to deal with sometimes. And, I think everybody should have at least some idea of a budget. So what I just generally do, I'm not going to go through this, but, I have a spreadsheet that I can do quick calculations on, like, what peak intensities I have, what 2-photon cross sections should be. In here, it's like, okay, here's what my wavelength is, my refractive index, power on sample, how many photons. And they're based on some estimates, like what the diffraction efficiencies, PSF volumes, based on the Born and Wolf correction. Look, I'm a serious guy, right? I use these corrections. But you can calculate all these things, and you can end up estimating how many pixels or photons you should be able to collect. And I don't expect these to be perfect, but I expect people, if they're microscope builders, or if you're attacking a new problem that nobody's ever looked at before, and you think, can I image at 10 kilohertz? You should be able to do a kind of a rough calculation and figure out, is this even feasible? If it's within even an order of magnitude, or even like a factor of 50, I'm like, okay, I'm going to do it. But if it's like 4 orders of magnitude off, I need to rethink my problem. Again, I need to rethink my task. Right. So I think it's important to think about this. If you're planning on building or doing something, you can get some estimates on the signals that you get and then figure these things out. And then, this again, goes back to these practical considerations for how to increase the signal noise. You need to collect, you need to do this. And these again go back into the photon budget, and you figure out what's going on. And then you try and make your experiment and you see, did I actually meet the conditions of my experiment? And if not, why did I fail? And is it a profound problem, or is it just that I had crappy alignment, right? Or my PMT bad? These are all things as builders we have to worry about, especially if we're pushing the limits of what we think we can do, right? If I'm just doing GCamp imaging with GCamp 6S and an easy region of cortex, and I just care about bursty signals, I can have a bad microscope and still do okay. If I want to do deep imaging or voltage imaging or like things like that, then I can't get away with it. So again, you should know your task and then think about what constraints you need to be able to solve your problem effectively. So, again, part of this is how to do the algorithms and what can the algorithms do to help us. So I'm going to go back to these NMF things. And these are these modeling structure. I'm not going to go through this. This is basically key. But, what you see in all of these is in the pixels, there's one thing that we assume. We assume that the source locations generally don't change over time. So if they change over time, then we have an intensity variation that's not kind of correlated to what we think the model is, which is a fixed spatial footprint of what our signal is. So one thing that's absolutely critical in almost all these extraction algorithms is motion registration. And the one that I use the most is, Normcore from Andrea and Eftikios. And it's important to use, basically be able to use rigid or non-rigid and whatever suits the problem. You calculate these things because these motion correction things are really critical for being able to do a good source extraction, because our model assumes that the kind of shape, the anatomy, the physical structure is not changing over time, at least on a short time scale, kind of an experimental time scale. So, really, if you're having problems with a lot of the algorithms, the first place to check is go back to motion correction and see, is your motion correction good? 90% of the time, it's motion correction failures, not algorithm failures, which is why when they said, what do you do if it moves too much? And I say, I cry. I mean, really, at some point, I cry because, your motion correction could be so bad that all of the, the best methods in the world can't salvage it in a good way. I have a habit of, after each data sets collected, and I tell the people I work with, kind of do a quick inspection of the motion, because if the motion is terrible, before you start imaging again or doing something else, you might want to take steps to improve that in some method before you go on. Otherwise, you could collect a bunch of data that turns out to be very, have very limited utility. So, what do we all do? Again, these are just refreshers. I apologize if you guys are bored with seeing these things already, but I think it's important to hammer people, right? Our goals are to record activity in the animal brain, right? And there's lots of imaging setups vary. There's lots of challenges and diversity in data. And there's a lot of trade-offs. We want fast, high resolution, cheap imaging, all these things. But the same culprits kind of lie across all data types. And there's noise, there's signal contamination, there's signal overlap. And what you see here is two volumes that we imaged, and this is dorsolateral striatum, imaged with, again, Rui Costa and some of his lab members, where we did 1-photon imaging, calcium imaging, through the same grid as we did 2-photon imaging. And here you see the kind of the two different panels, right? But you can see in the 1-photon imaging, you can see the neurons are the same as in 2-photon, but you have a lot of background, right? But 1-photon is way cheaper. There's a lot of photons. It's fast, right? Same thing here. Here's some of the little extracted components, again, showing matching. So whether it's 1-photon or 2-photon, 1-photon, we get the signals easier, but, and it can be faster, but we have more background, whatever. So again, this is just illustrating all these trade-offs. So what do we do in all of these cases is we really start looking at these models and say, what do we really have? And I think there's a kind of a detour that's a good one. And this is with, Liam, Ian Kinsella, and Amol Parsakar. I'm not quite sure why their names don't appear, but Liam's on top, Ian Kinsella and Amol are in a kind of, let's say, descending order on the page. And, all I'm saying to do, we have this model. Why? Well, why is the observations? It's, it has units of essentially pixels times time, right? And what we claim is that there is some number of sources, some number of locations, and that's this A matrix. A, there's some number of sources over these pixels. The C is this time varying signal, and then we usually have a background component that's an element, and there's some Gaussian noise. And a lot of times the background is low rank. So what do these NMF forms generally do? Andrea went through this very well. We try and solve this constrained minimization problem, usually an alternating minimization problem, where we try and solve for A, C, and B such that this is minima, this Y minus AC minus B in the L2 norm is as small as possible, subject to constraints that there's non-negativity in A. We don't think, or in C, we don't think the transients go negative. 0 is the lowest it gets, and whatever. And these things all happen kind of at once in some of these instantiations. Like in the first CNMF method, this kind of handled it once. the compression and demixing happened because we had a model underlying the type of spiking. It was an autoregressive model that described how things went up and down. And this essentially did some denoising and compression of the signals right on, and it was all part in ones. But now we're finding in some cases it's actually maybe sometimes better to do a multi-stage approach where we might do some denoising first to separate it from the noise. When with that denoising comes a compression, and then we can do the demixing part on the denoise signal rather than the kind of original noisy signal. And this has a bunch of advantages, both in speed sometimes and in the ability to detect weak signals. So, I'm going to show some denoising that's different from what Jerome showed. I mean, I think deep interpolation, that's kind of like the state of the art for denoising using kind of machine learning for these kinds of images. But, it requires some amount of training, and there's some things that have to go into it. And sometimes, we like having things that have maybe less training or less models for some certain types of things. So we have the kind of an alternative. And when I say we, it's really, Ian, Kinsella, and Liam on these. And that's using this penalized matrix decomposition approach. And this is essentially, you can think of this as kind of like a PCA approach, so principal component analysis, where you assign a little bit of structure, again, based on some priors of what you believe in the signal. So, our belief is that the signal should be kind of roughly spatiotemporally smooth. Also, the shapes, neurons shouldn't be there, disappear, be there, disappear, be there, disappear. There's some amount of realism, so to speak, in these neurons spreading across space. And we think that the activity should be kind of generally localized. So by having this model, the nice thing about it is there's really no parameter tuning. You can basically run it. It automatically generates parameters and makes estimates of these for the signal. And it seemed to work reliably for a very wide variety of use cases. And it's very, very quick. What does this look like? It looks kind of, you could say, similar to what Jerome showed, where if we take, let's say, a grid lens image and we look at an example, the average of the time series of frames, it looks pretty, let's see if I can get my pointer back. This is average time series focusing on this during a particular time frame. And the original data, we can see that the neuron is active for sure, but there's not much other structure. In the kind of compressed PMD denoise frame, this is what that frame would look like. So it significantly cleans up the frame. These sources become more highlightable, and it helps. I should note then, I'll go to the next slide. Like deep interpolation, it generates an increased SNR on the kind of small pixel number trace. So what you see here is now what the original data looks like on a 4 pixel average on the top left, and then on the top right is the PMD denoise of the same pixel. So there's some loss of noise, not all of it. But if we look at actually the region of interest average, the whole average of the whole cell you see in this yellow circle, it's actually the PMD signal, the compressed, doesn't look so denoised from the average. So what this is just basically doing is it's capturing the fact that as a cell, the ensemble of all the activity in the cell, essentially, there should be signal shared between it, so it can essentially distribute the ensemble noise and average across it over the whole ensemble, and put it in there, right? It doesn't do what deep interpolation does, which is to remove the completely kind of the noise that could be structured from other things other than just kind of pure little shot noise, which could be these kind of individual variations in background. So if you record the signal, and Jerome kind of showed a nice example of this, if he didn't tune the network right, you saw what looked like a little bit of beating in the background. That beating is essentially what you see in the background of these traces. So these remain here. So we're not really killing the super high frequency noise. And Jerome showed a graph of the PMD's performance versus deep interpolation. And indeed, they performed about identically for what I would call low frequency variation, but for higher frequency variation, deep interpolation was doing a better job of tamping the noise and going towards the ground truth, at least at the data sets simulated with Naomi that he used. And I should say this is kind of a little bit expected, because in this PMD structure, we don't learn anything about, let's say, the way that the neurons go or the way they fire or their shape or these other things. It's really a more unstructured model. The nice thing about this results in huge pixel compression, and it's very, very fast. So what we have here is we have an accelerated workflow where if I had a 25 gigabyte image, which is not, I have hundreds of 25 gigabyte image data sets, thousands even, unfortunately, probably a few thousand, where we would have to find this Y, we'd do this demixing, get ANC, evaluate, and we'd have to tune this. But this was very painfully slow. You couldn't really tune it interactively because it's operating on this giant movie. So now what we do is we kind of compress this movie into this reduced, this kind of sparse representation that has much lower dimensionality and rank, 250 megabytes. Now we can do this ANC tuning very, very quickly, fast, almost in real time, even for this movie, and then modify some of the fine-tuned parameters of the source extraction of the NMF algorithm to get these things out. So this works quite well, and it's nice. Again, it has less structure. I think deep interpolation, if you have heavily noisy images, you're sure about the structure, it works very well. But for things that you might not want to train on, or for compression and all these other things, PMD, and for this fast iteration, PMD also works quite well. We're exploring, of course, using deep interpolation also as inputs of this, and we like to borrow from other people's good methods. So I think you all should be doing that too. So one of the questions that came up earlier too is, well, okay, these aren't convex problems. How do you initialize? And how is this trouble? So with Liam, with a project led by Amal, they're coming up with different ways of initializing that incorporate some of these kind of AI segmentation, where they came up with this thing, mask NMF. And we already noted, if you imagine you had densely overlapping signals or very sparsely active sources, it could be hard. So what this does, I'm not going to go through all of it together, but it essentially just says, well, let me first just kind of like estimate these sources like as if they're not separated. I'm just going to like take, generally like ROIs, and then I'm going to look at the fluorescence, and I'm going to deconvolve it, so try and get events just on onsets. And then once you have those event onsets, you essentially multiply that convolution structure, which instead of having transients that go up and go slowly, you just have essentially sticks that it's bright or dark based on the onset. Now, if you then look across that projection, if you multiply that temporal matrix with these kind of spatial footprints, what you get is essentially a movie that a neuron is only active for like 1 or 2 frames, and then it's not active. You don't see the tail, and you get this projection that then shows something that looks like many fewer neurons are active compared to the raw trace, because it's only the neurons that kind of turned on in that interval. Now, when you do that, you can look now, do essentially a convolutional neural network and identify these frames. So now you just look at the brightest frames, and you use instant segmentation to identify the neurons, and you can train these things. And I'll try and show, let's see if I can click and get this video to play, where, what it is these are the subsequent frames in this kind of compressed movie, and now we can initialize spatial footprints. So these provide very kind of good initializers, again, based on the combination of kind of a little bit of sparsifying plus cleaning up, aided by a neural network identifier to do it. So, you can initialize by manually clicking. You can use these kind of tools to do it. There's many ways. We find this method works very, very well for 2-photon data. For 1-photon data, there's some different initializers that work. So, just keep your eyes open. But this works very well for these kind of things. And then none of these tools really mean anything if I'm the only one who can use them. So, like with John Cunningham and with Taiga Abe, they've developed this Neurocast pipeline. So this is neuroscience cloud analysis as a service software. So what this is just something that runs on essentially AWS, while instead of, like everybody loves Colab and Jupyter Notebooks, and I use them all the time, and Docker, at some point, like I didn't even want to use any of that. I just want to upload my data somewhere and have it work. So in this case, you just log into the website, you can select your algorithm, you essentially review some parameters, you dump your data sets, and then it spawns out AWS instances, computes it, and gives you the results back. I should say, the website is up. It just search for Neurocast, and it has some main analysis programs. You can see Cayman on there, but Deep Lab Cut, the Loca NMF, this PMD decomposition, right? These tools are on there. So if you're interested in trying out some of these tools, just reach out, go to the website, and there's information on how to contact them. And again, the main people on getting the Neurocast pipeline up and running would be like Taiga and John, And then, Liam, Ian are also very involved in the algorithm side. And there's many, many others, right? It's not that these people, you didn't see the full list of names, but this provides it. So now, again, it's reproducible. You can do it. You can share it with your friends, so to speak. You can get this data. So that kind of summarizes what I wanted. I see a question.
Hi, is this free?
Right now, it's free. There's part of a grant, right? At some point, what's not, at some point, storing giant amounts of data on AWS or running, it becomes expensive. And at some point, it'll transition probably to a model to use the user's AWS account. That's not the case now. But I would say is in terms of like cost, I've put some big data sets through it. And when I mean big, I mean like that, take kind of a few hours to run to a day to run on my local computer. And my local computer is a dual processor Xeon with 512 gigs of RAM, Titan, RXP, I have like the best computer that you could buy for a desktop computer. I still can't spawn out 100 things to 100 instances. So something like that ends up being like a few dollars to analyze a data set like that operationally. So I would say, considering at least in New York City or another place, like on NIH minimum postdoc salary, it still costs over $200 a day to pay a postdoc to $250, right? And if they're spending all their time trying to futz around getting the algorithm to work instead of spending $3 to run it somewhere else, I think it's a poor use of resources, I would say, if these tools work for you. So like even when it becomes kind of like, let's say, a fee, there's a fee. It wouldn't be Neurocast, it would just be the user who pays for the computation. I think it's a good strategy. And the nice thing about it is, again, you don't have, why Docker and all these things were invented is you don't have to worry about, well, which version of the library is it? Is it updated? Is it whatever? And it just, the goal is to have it kind of run. So with that, I just want to, I guess, do my imaging summary conclusions before I move on to control. I'm sorry, I'm not looking in the chat. So if people have some questions on this, I feel like, feel free to interrupt because I'm just not, I didn't know that. But one size doesn't fit all, right? Match the imaging strategy to the sample and the task. Figure out what you want to do, figure out what imaging would work, what you can afford, all of these things, and figure it out. And then I'd say we can gain by shaping the things that we can control, right? I can't control necessarily the fluorescence quantum yield of a fluorophore. I mean, there's protein designers who do design for that. I don't do it, right? So, but I can control the PSF. The other thing is all imaging really involves some estimation. So take advantage of the statistics that we know about, right? Like we know that neurons have certain compactness. We know something about signals, and we can leverage a prior knowledge of the sample. And to do this, then we can do these jointly designed systems where we kind of think about the systems as not just a microscope, but it's a microscope and the algorithms that we use to either generate the kind of light, the light field that we use for excitation and for data extraction. And all of these things actually combine a complete system that we normally call a microscope, right? For doing, for doing an experiment, it's not enough to just know what the microscope is doing. You kind of have to have it in that whole ecosystem. How does a microscope generate what I want? What's a microscope do, and how do I get the signals out? So that works well. So now I'm going to shift to my second kind of part of the talk, which is on modulation. And these modulation things is because, we're doing a lot of observational neuroscience, right, when we're doing imaging, which is great, and we can measure a lot of things and look at causation, and with good experimental design, with kind of controls, we can even start getting into the causation. But what we really want to know, in many cases, or at least what I want to know is, I say, oh, this neuron is doing X. Well, what's it really doing? Well, I want to be able to turn it on or turn it off when I want during a computation and see how it impacts the structure. So, our goal in all these cases is, the brain is in some particular state, and we want to be able to kind of progress. Sorry, I'm seeing if my pointer is not appearing on your thing. Let's see. I think PowerPoint decided to do autosave for me. You're going to have to bear with me for a second. But anyway, you can see this red line, right? Imagine each time the brain has a choice, so to speak, free will discussions, not part of this lecture, but in each choice, right, there's a certain path that it wants to it goes down, right? And with these things, let's see if this is PowerPoint's deciding it wants to be a little bit slow on me, right? But we want to be able to control this in a deterministic way. Let me just see if I can figure out what's going on with this. If you saw my computer, I'd be telling you the secret to the universe, but unfortunately that slide is blanked out. What I may do is, if this gives me trouble again, I may stop my PowerPoint and then start the slideshow again, and I can just jump to that because it's being very, let's say problematic with that. So let me just go to the slide that we want, blah, blah, blah. Do you see my screen again or no? Let's see, I'll get it. Screen 2, how?
Oh, it's gone.
Am I back?
Yep,
Yeah.
You're back.
Okay, great. Hopefully this won't be so troublesome. At any rate, we want to do this. So what do we do? You know, I like this movie because it's fun. So this was with combination with my lab and Rui's lab, and we used, Hillel sent out ST Chrome when we first used it, and ST6, and GCAM 6F in this case. So we want to be able to control cells based on their identity. So what you're going to see, there might even be some sound. When you see these bright lines appear kind of in this, like those lines that you see, that noise, I'm doing photostimulation, but I'm not targeting any neurons. Now I move my point to some neurons that I know are expressing opsin, and you can see that I'm turning them on. And, it's kind of cool that you can do these kind of things, right? Because now instead of just necessarily identifying neurons based on a genetic subtype or very coarsely, we can actually just pick cells that we want to modulate based on their functional identity as well. So this allows a big repertoire of activity. And again, I did some of this earlier work when I was with Rafa and with Wei Zhen, and we did, a similar diagram. I don't think we need to see it, but, you have a scanner for photostimulation, another scanner for imaging, and you can use a Spatialite modulator to basically define things in 3D. So what you're seeing is these are patterns in the XZ plane down here. So, what you're seeing is I basically got like a cuvette, filled it with dye, trying to put a camera on the side, and you can see it. So you can do kind of reasonable control in 3D, XYZ, and stimulate neurons. And, like there were some papers that came out from that, right? There's this paper on the cortical ensembles, and then we have a paper in eLife where we're doing, simultaneous imaging and optogenetics. So this all optical physiology. And I should say that I'm just showing my papers because I have these slides, but many, many other people at similar times were doing variants of this. The Heuser lab, Deiseroth lab, Chris Harvey. I mean, the list goes on, Hillel, and so on. So, what you can see is, here we can stimulate a huge number of neurons in 2P. This looks like, it's like epileptiform type activity, but, there's a 2P activation and you can blast the hell out of whatever you want. So we can do this. So like, what do we have to worry about? And, you've heard this referred to many times. This is an example of good data that helps move the needle, right? It's not a sexy problem, so to speak. People don't like thinking about it. It's not like, oh, this is going to the cover of Nature, right? But, it should have been in something like a Nature Methods, in my opinion, because it's a critical thing to understand how much power can you put on the brain and do imaging, because it really defines these limits, again, of what we can hope to achieve using certain strategies. So in this paper, they showed that, at least using these particular monitors for neural damage and kind of heat activation, that you could actually image with a fair amount of power or for a long time without seeing a lot of activation. That's not to say there aren't some subtle effects somewhere or whatever, but there didn't seem to be any gross effect. And I can say for myself, as behaviorally, we don't see any gross effects. If I was a synaptic physiologist who cared about detail plasticity or conduction velocity or something, I might have different concerns. But for like computations related types of behaviors that we do, in my lab and in many other labs, this paper really gave us some background that we could say, okay, we think we're in a safe regime, and here's why. We could cite, that this worked. So it was nice. The other thing that we did is, okay, given these constraints, how do we be able to stimulate much more neurons? Well, we use low rep rate lasers. And the low rep rate lasers are great because we can make the peak power very high while keeping the average power low. So instead of having an 80 megahertz laser, I have a 1 megahertz laser with the same average power. I have 80 times more intensity in a single pulse. Well, that 2-photon intensity would be 80 squared. So I have a huge enhancement of my ability to activate a cell or get 2-photon activation using these lasers. And that ends up being nice. You still have to worry about things like crosstalk with imaging, which is kind of what's shown here, which is kind of a dark secret that many of us don't always talk about, which is these opsins almost always have a blue shoulder. So as you turn up your imaging power, the number of spikes you get, you start activating the cell with your imaging laser. So we try and keep below the limits that Casper showed, but also usually we go lower than that even to get away from this point where these activity patterns really change a lot. The other thing you can do, and I mentioned Tommaso Fellini, right? He's really driving the use of the blue opsins with red indicators. Now that can be made orthogonal, but you pay a price because the red indicators are just very far in quality from the green indicators right now. And the lasers to get high-powered 900 or 920 or 800 are much more expensive than a high-powered 1030, 1064, 1080 laser. So, this all looks good. We're happy with all these things in theory, but then we start looking around and say, well, can we get single cell? And here we see for kind of targeting one cell in one plane, you can get something that falls off so rapidly, in like a few microns. It really looks to be single cell. As you start adding more and more targets, you see this kind of non-specific activation. And some of this is due to this was work taken with non-somatically restricted opsin. This is C1V1 expressed everywhere, not the modern opsins. But if I expressed a modern opsin with the somatic restriction, I would still see some non-specific activation if I have a lot of stuff going on. And I should say this isn't due to anything related to like my system itself. My targeting accuracy over these things is essentially sub-micron on average. But I still have these kind of fall offs with lateral distance, and especially I should say axial is always worse unless you're doing temporal focusing because you lose this confinement. The point spread function's oblong in these cases. But what's really going on? And part of this has to do with these ideas of saturation. So it's related to differences in opsin photophysics compared to fluorescent sensors. So this is a nice paper that, not exactly a barn burner in number of citations, but I think important. It highlights a lot of big factors, which is that, as we increase the power, if we have absorption, we undergo this thing called saturation. So the point spread function, even though the microscope hasn't changed, the effective optical kind of sectioning, it gets worse, it increases. And, normally this isn't a big thing with most fluorescence microscopes because we're not imaging that close to saturation, but opsins are a little bit different. And the reason that opsins are different is because opsins, the way we use opsins is very different from the way we use, sorry, my PowerPoint, oh, there it is, good. My PowerPoint recovered. But, if I take a fluorophore and I have a probability distribution function or like the PSF for a single shot, and the fluorescence relaxation time is much, much less than less than the laser rep rate. Each subsequent shot just basically is independent of the next, right? The fluorescence decayed, the fluorophores are there, it doesn't really matter. But with opsins, I run into this situation generally where the tau off, this is a channel opening time. Those channels stay open for milliseconds. So now my laser comes, some of it's excited, but then the next pulse comes before the rest of it get excited down. And then this effectively saturates the signal. And say this isn't, so to speak, rocket science, it's actually just basic probability. And it says that basically, if the probability, let's say I have something that the probability is only 0.1% that I'd be able to get this excitation per shot. But if I give it 100 shots at a 1 megahertz laser, right, that's 100 microseconds, it's not that long. The probability that I would get that absorption is 10%. if I did 1,000 shots, it's much higher. So, if it was 5%, you're at 94% or 99% activation when on a single shot it's 5%. So these excitation functions grow as you take the increase in number of shots and as that option is open. So these are sometimes hard to beat. So, this points to a strategy that you should try and open everything with just one shot. That points to using disks and so on like that, as opposed to spirals. But there's some other effects that play into this. It's not like nature never makes it quite that simple, which is if we compare the amount of heat you use for one versus the other, if you use the spirals versus disks, you use less heat in general, less total power is required to do the cell. And at some point, because of opsin photocycles, you might not be able to drive every opsin into an open channel if you only do a single instantaneous shot. So there's been some studies on this. We explored it in our kind of eLife paper. Valentina's, Miliani has done some temperature rising. Other people have done it. So, but again, it points to there's no clear winner strategy, I would say, between how you can do this. I would say for most applications, spirals work very well and they're simple to implement. But if you need very precise spike timing, you want to open. So when I say spiral, what we're doing, actually, I should go back and say, what we're doing with a spiral is, I'll go to annotation, right? Where if we have a cell, imagine the cell of this panel, we're painting the laser on with a small spot like this. So I think, do you see the green circle on here? Yeah, so you're painting the cell. So this is a temporally multiplex strategy in a sense. I'm moving the point to paint it. This takes some time, but we can do it in a few milliseconds. It's with modern options, but it still takes some time. On the disk strategy, what you're doing is you're making a light field or a light pattern that covers the whole thing at once. If you activate it all at once, all the channels open at once, so you have a much bigger influx of instantaneous current, and that can drive spiking more reliably. So if you need the sub-millisecond jitter, disks are really the only way to go. But if you can handle a few milliseconds or a couple milliseconds, the spirals also work. So again, for 95% of applications, I think either could work. That's a take-home message. Both can be good. For the marginal cases, if you're at the limit of total number of cells, the power efficiency for spirals might win. If you need absolute precision and spike timing, you might want to go with disks, right? But they're kind of, I would say only semi-complementary because the biggest part of their thing is overlap. So, with all these things, there's a bunch of things to do if you're doing real experiments, right? So you need to figure out actually, if I said you can't do single cell easily, there's a bunch of work trying to use optogenetics for mapping. I'm not going to show almost anything on this except to say that if you're using optogenetics to try and do mapping of cells, you need to understand how these biases affect your populations and how to do it quickly. And there's a lot of great work with Liam and Hillel and Marcus and Marta. And I just talk to them about it. I'm not really involved in it, but I know what's going on. And I think it's important to put out there that, depending on tasks, you really need to, again, think about jointly how I'm going to extract a signal, how do these biases play into my experiments, and put all these things on to really do it. The other thing I'd really note is that there's a ton of new and better options. So if you're just getting into this field, man, it is fantastic. So Hillel has some great ones, the Deiseroth lab, the Weigert and Hegemon labs. I mean, they're getting more sensitive, more redshifted, more orthogonal, really it's changing what you can do. So just so I don't, kill you all on time, if you can't always get single cell, well, you can get very near single cell. So here's basically a response pattern to a single cell in vivo, looking at calcium responses as a function of these grids. So we're scanning about a 10 micron spiral, and we're shifting it by 5 microns, and you're seeing the excitation pattern. In this part B, you're seeing kind of what the calcium responses are at each of these points. And then in C, you see this target stimulation as a function of distance. And if you remember, the spiral is 10 microns. So when we shift it over 5 microns, it's still half on the sample. By the time we're at 10, we have very little spike probability. So we can get kind of near single cell. I would say certainly we can get single cell precision in targeting, but whether we excite only that cell, that's the challenge, right? We can definitely excite the cells that we target, but we also probably get a couple of cells that are nearby if they have high opsin. And that's just because the cells have membranes that are touching each other with a kind of nanometer spacing. Unless you're, optical resolution can't fix that problem. But they can get near single cell, right? So what you do when you get this, well, what can we do? These are some of my team members with Rui as well, and Nuria was with Jose Carmina. We were using this option, and we looked and did a bunch of stimulations, and we said, well, what kind of precision can we get? And we did, this is the GCamp 8S in this particular case. Sorry, no, actually, this is GCamp 6F with 8 seconds. I had some other slides where I used a new GCamp to do it, so I apologize, but there's the 8 seconds. And you can see whenever we stimulate the cell, what you're seeing down the line is kind of the fluorescence transients and a bunch of trials. And each one of these, there's a success. We don't see a single failure in hundreds of trials over many, many sessions over the past year, right? It has very good stability. And if you look at like a nearby cell that's right there that also has optin, we don't get activation. So it's kind of quasi-single cell. And we can pick other pairs and do these kind of things. And what we see is, you can see a little stim artifact in the other cell, but you don't get response. So this is really cool. But what can you do with just a single cell stimulation? I've been talking about all this SLM stuff, so I want to highlight, I think, a really cool experiment that I worked with Attila. This was mainly work by Sebi Roletti, who's now at Control Lab or Facebook Reality Labs in Attila. You can see the long list of authors. This paper kind of just came out. It says in the future, but it's out now, right? And we're looking at local inhibition. So like how place cells are formed, there's lots of hints that there's kind of this strong feed-forward local inhibition that then is controlling kind of place cell formation. So if you have to have essentially sparse activation of the pyramids with kind of these dendritic spikes to activate place cells. And if all the other cells are active around it, surround inhibition will get rid of it. So in this experiment, we said, well, with single cell electroporated a particular cell in hippocampus. And as the animal was making its rounds, we saw there was no place cell. This is one of these patterned treadmills with lots of rich cues. We did some laps and we did some stimulations, and we can see after we run the stimulation period, we can induce a place cell, and that place cell is reliable for at least a day. It really works, and you can see here in the induction times, right? There's some other big things, and it works pretty well. So this is a single cell photostimulation. Now, the thing that was interesting about this, if we expanded our stimulation, and so if we expressed the opsin more broadly and we did single cell stimulation, you could still do it. But if you expanded the stimulation to include more pyramids around it, the induction would fail. So it really was a nice hint, and you can see this in these experiments that when kind of the density of stimulation was high, the tuning distance, the ability to tune it to the kind of the precise field, the shifts that we got were much, much smaller. So this is just a very small subset of these experiments. There was wide field experiments, many, many others. But what we were able to show here was just doing kind of single cell versus kind of like bigger cell patches, that in here it really looks like it's a kind of strong feed-forward inhibition that comes from recruiting other pyramids, and they recruit the interneurons, which then suppress the formation of play fields. So in these cases where we did this kind of medium or higher density stimulation, meaning we activated more cells in an area, if you dreaded out the interneurons, you could have induction of play cells, but you needed to dread it out. So it's these kind of looking at these kind of local inhibitory circuits to figure out what's going on. I don't have much time left. I usually don't talk so much about the science in these things, but I think it's important to show that with a good design, even doing single cell stimulation, if you pick a good experiment, if you have a good experimental control and design, you can do really cool experiments. So if you don't have an SLM, you don't have a bazillion-dollar laser, even with like a normal laser, one cell, the ability to control one cell precisely versus a few cells can be game-changing for some experiments. The other thing that I work on a lot of my time on is this giant U19 award that we have with many people here to try and understand motor control. And motor control is a very complicated thing. M1, there's many areas involved in cortex and basal ganglia, brainstem, cerebellum. All these things are interacting. So what kind of our hypotheses are in doing these kind of experiments is we really think that the neural activity, let's say the neural activity in M1, it both underlies issuing the output command. So there's information in there that's encoding the action in some way, but there's also activity that's important for the dynamics to transition to the next activity pattern. So if you think of it this way, right, there's functional characteristics of the neural circuits, and that underlie structural connectivity and these inputs. And you can't almost like how like with a hologram to Nico, like if we have something at a time point here, it might enforce a certain kind of activity pattern up or downstream if we need it to exist in a certain way. Just like this kind of propagating hologram, there's this idea of dynamics as this time propagation. So we think that there is this issuing a command, there's a downstream readout and output, but we also think that this embedded in these activities also a transition to the next activity. So ignore the details of these slides, but the basic point is that both of these structures have to exist for the brain dynamics to work. There needs to be underlying dynamics to sustain activity in the next motion, and there's these command structures. So what do we of course want to do is we'd like to be able to drive particular sequences and dynamics and test whether these things exist. So we work on things to drive arbitrary patterns of activity. Here's a very simple one. We have 4 cells. Kind of, if I play the movie, let's see if it plays. You can kind of see this triangle. If you kind of look around and know where those arrows were, you kind of see these time series. If I actually look at it, and we now just plot the calcium activity, right? We can see we activate the cells when we want in the sequence, and this. I mean, it looks good, right? This is of course very slow activity that we're injecting in here, not related at all to motor timescales, but just kind of it helps us visualize it, right? So now what we want to do is we want to basically have algorithms that we watch activity when the mouse is doing a task, a particular motor task. We can kind of infer what the dynamics are, and then we want to perturb them in meaningful ways and both look at the network activity or the overt changes in motion as this goes to test some of the hypotheses of kind of which neurons are kind of coding the direction, which neurons are coding the transitions. Are the signals mixed? Are they really dispersed and whatever? So to do that, we've been doing a lot of work on showing that we can get very tight sequencing and triggering. So here's just an example of doing 30 hertz imaging. This was, GCAMP, this was a 6M in this particular case. This was a Carmine construct bicistronic with 6M. And these are just delays between 4 cells that we chose A, B, and C, and D. And we said, let's try it with 180 millisecond, 120, 60, and like no delay between these. And these are average of 9 trials, but the same results are faithful for that. And you can see we can very precisely control the relative latencies between these cells down to the order of within a couple of milliseconds, a little bit, maybe even less than that in these cases. So now by developing these tools and the controls and verifying that we can actually stimulate on precise sequences, we're trying to do these experiments where we're doing these closed loop experiments where we want to trigger photoactivation to achieve some ensemble activity and see that this thing works. And we've done some preliminary experiments. This requires a lot of software where we can show that we can kind of shift the activity of one ensemble and not another. So I mean, I'll spare the details, but when we do stimulation, we can see this is a stimulation. We can move the activation of one while not moving the other. So this is kind of what ensemble 2 is doing, and ensemble 2 within and out of stim. What E1 is doing, that's a non-chosen ensemble, changes a little bit, but not much. And now this is the E2 minus E1, and we have a calibrated threshold for the stimulation. And we can kind of move the activity of these ensembles in precise directions where we want it to go, kind of based on our models of what we think the dynamics are. And with that, I'm coming close to a close, like to do this really well, we can't just do motor cortex, so we do imaging in grin lenses. Grin lens imaging has actually striatum is nice for imaging. The raw movies on the left and just background subtracted movies on the right. Striatum is pretty silent until you do motion. So you get these very, very high SNR movies, and you can do it. But of course, it's kind of invasive, but you can follow all kinds of cells across imaging and learning. Inej has an isometric force task that the mouse has to learn. Isometric force tasks are hard for a mouse to learn. And we can look at the same neurons over days and weeks as it's learning the task. We can figure out what basically cells are D1, D2, how they change around. And now we're getting to the point where we want to now stimulate them during the course of it. So I'll just finish up with my last couple slides, which is now we're starting to really push stimulation through grin lenses with single cell. So what you see here in panel A, this is this average frame. Panel B is the raw frame, and panel C is a denoise frame using PMD denoisers. And then we do the NMF to extract out the source. And there's a little source viewer showing just the cell being that gets activated by the stim. It's only one cell picked up in that component. And you can see the traces below. In this one, we're stimulating a cell with 4 activations. 300 milliseconds apart, and in each one you can see the things going up. And I should say this is repeatable for different mice in the field of view. Here's another cell where we did actually much smaller stimulations. It's kind of all the neurons. This is showing all of the neurons that were extracted for running NMF. This is just a cell of interest, and I zoom in on these traces. And again, here we see these just 4 precise patterns repeatable over time. This is actually using GCamp 8M with Carmine. And now our big goals is, okay, now we can stimulate these things, we can do it. And again, we really need to kind of close the loop. So with that, we're trying to do all of these things, utilize Cayman, Oasis, all these tools, and put them together to do kind of closed loop behavior. This is actually very hard to do well. We have to do the network fitting, all these, and we're really trying to do it. So I would say the conclusions on the control is that we can stimulate lots of cells. You can do this all optical physiology, and it's pretty flexible because you can identify cells by genetics or function. But again, with this great power comes great responsibility. You know, technology isn't going to solve all your problems. You really need good experimental design that takes into account the biases of both your measurement, your photostimulation, expression, and all these things to do it well. And I should say, you know, overall, I'm very positive about these things, and like all these strategies are increasing fidelity. And one of the challenges for me is to kind of not be careless and be like, yeah, we can stim anything you want, but to say, yeah, we can probably stimulate anything you want, but we're also going to stimulate a bunch of other junk maybe, right? Is to kind of communicate this really effectively. And with that, I'll just close it out and say thank you. And thank, you know, these are the main people that I work with on some of these things, but of course, most of what I do is collaborative, so it really involves many, many other people. As I say, it takes a village. So I, you know, and then our funders, and I, you know, thank you for all for your time.